There's a type of planarian flatworm that's a relative weirdo among its fellow multicellular creatures. This planarian flatworm is a weirdo because it has a special ability that very few multicellular creatures have, and of those who have this special ability, almost none of them are as effective with it as the flatworm. So what is this special ability? It's the capacity for the multicellular organism to regrow its own tissues. And I don't mean just healing from a small wound like a laceration or a puncture. I mean regrowing maimed limbs, or regrowing an entire part of the body that's been severed off. A lot of creatures have this rudimentary ability, including some vertebrates, like various species of lizard and salamander. But the flatworm is the weirdo, because the flatworm can regenerate itself to an extent that virtually no other species can match. Where some small lizards and amphibians can jettison their tail as a defense mechanism and then they'll regrow their tail over the next several weeks, the planarian flatworms can regenerate any part of their body. This incredible ability makes the flatworms a very exciting subject of research, particularly medical research into tissue regeneration. A recent study into the flatworm was published in Biophysical Journal, which made a fascinating discovery about the earliest processes of tissue regeneration. The study conducted experiments on the flatworm species Dugigia japonica by removing parts of the body. They cut the flatworm individuals into chunks, and they performed an intensive analysis of the chemical processes that led to the regrowth of the removed tissue. Now, before the research was conducted, it was already known that particular genes associated with the regrowth and regeneration of the injured tissue would become active about 6 hours after the injury. But until now, what was happening during those first 6 hours before the genes were activated was largely unknown. In the introduction of their paper, the researchers begin by explaining the complexity of the regeneration challenge, saying, quote, "Regeneration requires the reconstruction of complex anatomical structures and their appropriate integration with the remaining body via precise control of scaling, position, and organ identity. Planaria are free-living flatworms that have an incredible ability to regenerate missing tissue after damage and amputation, despite having a rich set of internal organs, 3 body axes, and a complex brain and central nervous system, all of which must be recapitulated each time they regenerate." Unquote. They're basically saying that the planarian flatworm is a complex organism. It's got a 3-dimensional body structure. It's not like a tiny little microbe. And it even has a complex set of organs. And all of these organs have to be organized and oriented properly so that they can all work together to keep the thing alive. So the mystery is, how does this happen? Because that's really complicated. The authors then explain how specific genes, specific enzymes, and specific RNA activity all interact to regrow the right kind of tissues. If the flatworm's tail is maimed, for example, it will regrow a tail and not another head, or a bulbous protruding gut section where there shouldn't be one. So if I could boil down this research to a single question, it would be, how does the flatworm's body know which genes to activate so as to regrow the right tissues? The answer, as the researchers discovered, has to do with electricity, or the electric potential of the cell's membranes. The researchers already understood from previous research that membrane voltage differences between cells in the tissue that's been damaged, these differences existed a day after an injury occurred. And so they hypothesized that these membrane voltage differences, quote, are in fact established and operative far earlier, unquote. Basically, they think that these voltage differences appear far earlier after the injury has occurred, and that they just might play some role in controlling which genes get activated where. To test this hypothesis, they took some flatworms and cut them into fragments at specific points along the body, creating injuries from which regeneration can be observed. They used voltage-sensitive dyes that would stain the flatworm tissue a particular color, depending on the regional membrane voltages. One of the earliest things that they saw was that the anterior ends of the cut body segments, or the end of the body segment that was originally closest to the head, it showed much more depolarization than the posterior ends of the cut body segments, or the ends that are closer to the tail. This difference in the cell's membrane voltage potential was evident within the first hour after injury, long before any region-specific genes were activated. This is where the researchers really start to fiddle with things. The authors say, quote, "To determine whether the early bioelectric state of planarian tissue is functionally important for proper establishment of AP polarity after regeneration, we sought to directly alter the resting potential of cells," unquote. Basically, they want to try and mess a little bit with the electrical activity quickly after an injury to see if that altered electrical activity will then alter the regeneration processes that follow. And lo and behold, it did. For example, in one sub-experiment, they soaked the flatworms in a potassium solution for a few days before they induced an injury, before they cut the flatworm's body in sections. In the days that preceded the injury, the potassium solution soaked into the flatworm's tissues and altered the membrane potential of its cells to the point where, when the flatworm was cut, where it would have grown a tail, instead, it grew another head. By manipulating the membrane potentials in this way, by making the tail section of the segment really depolarized in a way that would normally produce a head, they were able to produce two-headed flatworms. They explain their findings, saying, quote, "Transient alterations of V sub mem, which are only applied for the first 3 hours after amputation, permanently impact subsequent gene expression and anatomical patterning events," unquote. So, to summarize, the membrane voltage potential of the flatworm's cells seems to operate like a biological map of the body. The relative positioning and orientation of these voltages seems to coordinate and help map out the subsequent gene activity that regrows the right kinds of tissues in the right places. And by messing with these voltages on the cell membranes, we can alter the subsequent gene activity, and thus we can alter the tissues and the organs and the structures that are regenerated. And that is so cool. Michael Levin is the senior author of the paper and the director of the Allen Discovery Center at Tufts University. To celebrate his team's work, he tweeted, quote, "We found that endogenous electrical signals enable cells to communicate and make decisions about their position and overall organ structure, so they know which genes to turn on," unquote. In an interview, Mr. Levin was asked about the medical application that this knowledge might have in humans. He said, quote, "With perhaps the exception of infectious disease, the majority of problems in health and biomedicine hinge on understanding how cells get together to build a specific organ or other structure. If we can figure out how to manipulate these processes, we can start to develop ways to correct birth defects and address everything from traumatic injury to degenerative diseases, aging, and cancer," unquote.